Well, it's 10 minutes on top of the hour. We are just 8 people here. I think we can't wait any longer. We just have to start the lecture and hope that the others join later. As at when they join, then they can also benefit from today's meeting. So before we start, I want to be sure that you can all hear my voice and you can see the screen. So you can, put up your hands if you can see my screen and you can hear my voice. So we confirm that before we continue. All right, great. So, okay, good. It means everything is fine, so we can continue. So that's okay. You can put your hands down. Thank you for that confirmation. So we are continuing today with our lectures on enzymes, enzymology. I think this should be the last lecture on enzymology, the general fundamental aspects of enzymology, so that from the next lecture, we should be moving on to specific, processes of metabolism. That is, we'll go into metabolism of biomolecules such as carbohydrates, proteins, lipids, and nucleic acids. We'll be taking each and every one of them and look at the processes of their metabolism. And we'll see specifically enzymes in action in the biochemical reactions that are involved in those metabolic processes. But today we are going to look at coenzymes into much more detail. At the beginning, we mentioned what coenzymes and cofactors were. And we mentioned that these are prosthetic parts or non-protein parts that when they are attached to the protein part, that is the peptide, which is the enzyme, actually activates these enzymes to catalyze the various reactions that they are involved in. And we mentioned that in some biochemistry books, the terms cofactors and coenzymes are used interchangeably. But in some others, you'll find that these terms are actually distinct from each other. And I mentioned to you that in that case, cofactors are referred to as the inorganic prosthetic parts of the enzymes that are necessary for the activity of the enzyme. So those are the metal ions that are needed for these enzymes to be active. Examples are mentioned to be iron, calcium, magnesium, potassium ions, etc., etc. Then I also mentioned that in that case, coenzymes are organic components. And some of those organic components are what we are looking at today. So we'll be looking at specifically about these 6 coenzymes: thiamine, pyrophosphate, nicotinamide, nucleotides, flavin nucleotides, coenzyme A, biotin, and folate. We'll be looking at their structure and their functions, their specific reactions that they are engaged in. So we'll look at the enzymes that they are coenzymes too. So we'll take each of these coenzymes, we'll look at the structure. And in some cases, we'll be looking at the mechanism of action of these coenzymes in those chemical reactions that they are involved in. Right, so let's start from the first one, which is thiamine pyrophosphate. Now, first thing you see is that there is this structure on the right-hand side here, and this structure is thiamine. Thiamine, which is commonly known as vitamin B1. And as the name of the coenzyme suggests, thiamine pyrophosphate is a phosphorylated vitamin B1. In that case, we have two phosphate groups that are attached. So if you look critically at the structure of thiamine, which is vitamin B1, and you look at the structure of thiamine pyrophosphate, you realize the only difference in these two structures is that you have these two phosphate groups replacing the hydroxyl group. The terminal hydroxyl group that you see here is being replaced by the two phosphate groups. All the other parts are the same. This ring is what you see here, this ring, and you see the five-sided ring is also what you see here, the five-sided ring. Now, what you need to understand is if you see the word pyrophosphate, pyro only means to die. So, in fact, thiamine pyrophosphate can also be called thiamine diphosphate. So, when you are reading, biochemistry and you see this structure that is being named, something pyrophosphate, know that the pyrophosphate means diphosphate. You can even have, say, something which is a disulfate. That can also be said to be pyrosulfate. So pyro means di, it means two. So two phosphate groups attached, thiamine pyrophosphate. Now, deliberately, we have this carbon and this hydrogen in a different color, red color, or call it magenta. That is because this carbon number 2, in what is referred to as the thiazolium ring. This ring is called the thiazolium ring. And that is because this carbon and this hydrogen are the reactive parts of the coenzyme thiamine pyrophosphate. Just like when we were talking about the active center of enzymes, we mentioned that you have some 2 or 3 amino acids that are acting as the specific portions of the enzyme that interact with the substrate upon which the enzymatic reaction is occurring. Similarly, you have for this coenzyme, thiamine pyrophosphate, this specific portion, this carbon number 2 and the hydrogen on it, as the reactive parts, as we will soon see when we are looking at the mechanism of action of this coenzyme. You realize that it is this particular carbon, this particular hydrogen that are involved in the chemical reaction that they are being coenzymes to the enzyme that is catalyzing those reactions. Now, you see the term thiazolium. Now, thiazolium means that it is, if you are reading biochemistry and you see a term such as this, THI, sometimes it will be THIO, thio, or you see thia or thia. It means there is sulfur present in that compound. So, for instance, if you take amino acid with sulfur in it called methionine. So you realize there is that term there because of the sulfur. You have that term. Then you also have in chemistry compounds containing nitrogen. So nitrogen-containing compounds are also referred to as azo compounds. And so you see that is where the term is coming from, thiazolium. So having both sulfur and nitrogen in this ring, and so you have this ring being referred to as a thiazolium ring. And that thiazolium ring is the reactive part of the coenzyme thiamine pyrophosphate. Specifically, the carbon number 2 and the hydrogen on it are those the reactive portions of the enzyme, the coenzyme thiamine pyrophosphate. Now, let's move on and get to see the enzymes that the coenzyme can be this coenzyme can actually the enzymes that you have thiamine pyrophosphate being a coenzyme too. So we have a table here where we have the first column, we have the enzymes. So the enzymes that we have thiamine pyrophosphate being coenzymes too. Then we have the second column where we have the chemical reactions that you find these enzymes, catalyzing, these enzymes acting in these particular chemical reaction pathways. And then the third column, we are looking at, the specific bonds that are being broken in the various reactions. So those bonds are in a different color, also in magenta. You see that bond there, you see this bond here, and you see another bond here. Those are the bonds that are being broken in the reaction. And we also have on the fourth column, you have the bonds that are formed. So after some bonds are broken, then new bonds form. We have those bonds also shown in magenta, different color. Now, what do we see? We see that the very first enzyme here is pyruvate decarboxylase. Pyruvate decarboxylase. And as the name suggests, the substrate in that chemical reaction is pyruvate. And the action that is being taken upon it is the removal of the carboxyl group. So that carboxylic acid group which you find, so this carboxylic group. So if this is a substrate, pyruvate. So if this was pyruvate, this R1 will be CH3. And so this will be the carboxylic acid group, the carboxyl group, which will be removed. And that is where the term decarboxylase is coming from, removal of carboxyl group. Now you find this reaction, ethanol fermentation. I mean, this is some anaerobic metabolism of carbohydrates. So the breakdown of carbohydrates, when you get to pyruvate, when it is in an anaerobic condition, you have the formation of ethanol. If it was some aerobic condition, you have the pyruvate after decarboxylation, then you may be having some conjugation with coenzyme A, and you'll be having acetyl-CoA, and then that acetyl-CoA, of course, will be acting in different biochemical reactions. Now here we see that in this process, this bond, which is between the ketone group and the carboxyl group, is what is broken. And you have a new bond which is being formed. This time around, it's a CH bond. So a hydrogen is replacing this carboxyl group, which is broken away. So this carboxyl group is broken away and given out as carbon dioxide in that metabolic reaction. And then you have the formation of an aldehyde when you have hydrogen being bonded on that carbon. So you have a formation of an aldehyde, whilst you have the breakaway of a carboxyl group, which is released as carbon dioxide in that chemical reaction. Now you also have an enzyme pyruvate dehydrogenase, which is involved in the synthesis of acetyl-CoA. So as I mentioned earlier, if it's still just in that process of carbohydrate metabolism. So it's the same. What is happening here? Bond is broken from pyruvate, and you have the carboxyl group removed from it. But in place of, you see here, you have hydrogen that is bonding on that carbon to give you an aldehyde. But here, you are having coenzyme A that is getting bonded onto that acetyl residue to give you acetyl-CoA. And that is why it's being referred to as synthesis of acetyl-CoA. Remember, I mentioned that when you see the word synthesis in biochemistry, it also means anabolism. That is the build-up of bigger molecules from smaller molecules. So you will see a term such as biosynthesis. It only means the build-up of a bigger molecule from smaller molecules in biological systems. And that is what we refer to as anabolic metabolism. See, similar reaction is occurring with what? Alpha ketoglutarate dehydrogenase. Again, the names dehydrogenases are the same as oxidoreductases. So what it means is that these chemical reactions which are occurring are oxidation reduction reactions. And you find these also occurring in what? The citric acid cycle, what we refer to as the TCA cycle or the Krebs cycle. You remember, even we'll look at it again, the TCA cycle. So you will see that one of the intermediates in that TCA cycle is what? Alpha ketoglutarate. So in that cycle, in that reaction cycle, you see such reaction occurring with alpha ketoglutarate, where you have carbon dioxide being given out, and coenzyme A is getting conjugated to the acetyl residue to give you acetyl-CoA in the synthesis of acetyl-CoA. Then you also find in the process of one of the pathways in carbohydrate metabolism is the pentose phosphate pathway, which through which you have the synthesis of nucleic acids. And you have also, the process of carbon assimilation reactions, which you see even in protein metabolism. And the enzyme which is involved in those processes is what is being referred to as transketolase. So the name tells you that it's a transferase, that enzyme is a transferase. And what it is transferring some ketone group. So, or can say it's a ketone. It will be maybe some keto acid or a compound with some ketone group in it. So now what you see is that, yes, the bond which is being broken, again, is between this ketone group and then this one carbon with some hydroxyl group on it. And there is a new bond which is formed after the old is broken. Now, this is not the same as this because the chain here is labeled R4, but the chain here is R5, which means these are different residues that are, so that the residue which is broken off is different from the residue which is getting attached. Now, this is not the same as breaking off R4 and then bringing R5. No, actually, the bond which is being broken is what is close to the ketone group. When a new bond is being formed, it only happens that, yes, still the carbon which is bonding back also has a hydroxyl group on it, but the rest of that chain is different from the chain which was originally on the carbon which is broken away. So you see, the thiamine pyrophosphate as a coenzyme is taking part or is participating as a coenzyme to enzymes that are involved in the breaking of bonds that are adjacent to carbonyl groups. And what are carbonyl groups? So the ketone group is a carbonyl group. Carboxyl group is a carbonyl group. So these two groups, you have these bonds that are adjacent to them. They are close to them, so they can easily be broken. Similarly, you have a ketone group. You have a carbon with some hydroxyl group on it. That bond can be broken by the enzymes that you have thiamine pyrophosphate being a coenzyme too. So you see these processes, you realize that you have decarboxylation, removal of carboxyl group as carbon dioxide, which comes out from pyruvate. And then you also have, the breaking of the bond, which is close to the ketone group here. And then you have a different group, which is bonded back onto the residue, which is left after that bond is broken. So these are the kinds of reactions that you find thiamine pyrophosphate, participating in. Now, so we're looking at the mechanism of action of thiamine pyrophosphate. Now, before I explain this mechanism of action, I want you to know that this is not something that you are required to be cramping into your head for remembrance. No, it is for understanding. Biochemistry generally is about understanding. It's not about, cramping things in your head to remember them, things like that. Of course, this whole thing over here, you can't say you want to cramp all of. If you want to be cramping every single, chemical reaction in biochemistry in your head, that is going to be very daunting. But it is for you to actually appreciate and understand what this whole process of the coenzyme is involved in. So I mentioned earlier that you have the thiazolium ring as the reactive part of that whole structure of thiamine pyrophosphate. So if you look at this structure, this is the thiazolium ring that you find in the full structure of thiamine pyrophosphate. So you have the part at the left-hand side being represented with R, and the part at the right-hand side being represented with R prime. So to appreciate what we are looking at, let's go back to the full structure of thiamine pyrophosphate before we come back to this. So what I'm saying is, this is the thiazolium ring. This thiazolium ring has some parts at the left-hand side. So all of this part at the left-hand side is represented with R. And all of this part, the two CH2s with the two phosphate groups on it, all of this part is being represented with R prime. And you are left with just the thiazolium ring, which is being represented in that reaction. Because, of course, we mentioned that the reactive part of the coenzyme is this carbon 2 and the hydrogen on it. This is the reactive portion of the coenzyme thiamine pyrophosphate. That's why we are representing only this in that chemical reaction mechanism. Of course, if you want to be representing the full structure of it, that would be so clumsy. We need only the reactive part. So this is what I'm saying. You have the thiazolium ring, you have R, which is the left-hand side, and you have R prime, which is the right-hand side. And you are left with just that carbon, which is reactive. There was a hydrogen, but the hydrogen ion is given out as a proton, or let's say as a hydride ion is given out. And so you now have thiamine pyrophosphate carbon ion. That's what you have. And by that, this carbon ion can have some nucleophilic attack on the substrate pyruvate. And that results into some temporary bonding between that carbonyl carbon and the carbon number 2 of the thiamine pyrophosphate. And you have the carboxylic acid group, which is to be broken away. That bond is broken, and you now have carbon dioxide coming out. You are left with that carbon ion of the substrate pyruvate, which is undergoing some stabilization, resonance stabilization. There's some transition states. There's some reversible transition states which could be occurring. just to stabilize the carbanion such that that carbanion can then, again, when it gets protonated, there is that protonation that goes on, and you have so that carbon which lost the on which you have the carboxyl group being lost from, and is carrying some negative charge on it as a carbanion, that now accepts this proton, this hydrogen ion on it. And you are now having this hydroxyethyl thiamine pyrophosphate, well stabilized. And then out of it, you can have that process of oxidation occurring on this hydroxyethyl to now have acetaldehyde. So what you see here is that this hydroxyl group, has been oxidized and is now a C double bond O group. This hydrogen still remains, and you now have that acetaldehyde. So that process of, what we saw in the table, that from pyruvate, you have decarboxylation, carbon dioxide coming out, and then you have the formation of the CH bond, where you have now acetaldehyde and aldehyde being formed. Step by step is what you are looking at. In the table, it was just shown that the substrate was pyruvate, and then there was decarboxylation, and you have acetaldehyde. But here you are seeing there is a step-by-step process by which this occurs. And this is showing how thiamine pyrophosphate, actually acts as a coenzyme in such a biochemical reaction. And once acetaldehyde, that is the product, is dissociated from the coenzyme, you now have the free coenzyme that can go on to the process of that, decarboxylation and then the formation of acetaldehyde from another molecule of pyruvate, and the cycle continues on and on and on. Now, I usually liken this process to, say, for instance, if you were in the kitchen and you want to chop some vegetables, and the vegetable was floating in the air anyhow, it would be very difficult to take your knife to chop that vegetable. But if you have some chopping board, and you place the chopping board, on a table, and you put your vegetable on it, I mean, that will be very stable, and you could easily use your knife to chop the vegetable nicely on that, table, and the chopping board, stable on it. Now, in this analogy, you have the coenzyme, thiamine pyrophosphate being that chopping board on which you have the substrate, which is, say, the vegetable, getting stabilized on, and then you have your knife being the enzyme, which is actually acting. So the enzyme in this case will be what? Pyruvate decarboxylase. Pyruvate decarboxylase as the knife, which will now act on the substrate, which is pyruvate. So I hope with that analogy, you'll be able to, remember, understand what this whole process is about, with coenzyme thiamine pyrophosphate, acting to stabilize the substrate for the enzyme pyruvate decarboxylase to be acting upon in that decarboxylation reaction, and finally the formation of acetaldehyde from the substrate pyruvate. Right. Now, the next coenzyme is nicotinamide nucleotides. Nicotinamide nucleotides. And this is coming from, so before we look at this full structure of the nicotinamide dinucleotide, this is the structure of nicotinic acid, or niacin, which is also referred to as vitamin B3. Now, take note, the first coenzyme which we looked at was thiamine pyrophosphate. And that was a derivative. It's a phosphorylated thiamine, which is what? Vitamin B1. We have jumped to vitamin B3. So take note, you will soon realize that all these coenzymes that we are looking at are vitamin Bs, vitamin Bs, vitamin B something, vitamin B something. So we'll come back to that. So you see vitamin B3, which is niacin or nicotinic acid. And this is the structure, nicotinic acid. Of course, here is written in an ionic form. You have this O with, carrying some negative charge. But of course, we could write it as in a molecular form as COCOH. That will be carboxylic acid. So it's a carboxyl, that's a carboxyl group. So you have niacin, nicotinic acid. And just in place, when you have the OH group, which is replaced with an amine group, then you have what is referred to as nicotinamide. Right. Now we see that nicotinamide here. Now, remember what nucleotides are. Again, from first year biochemistry, we knew what nucleotides were, the basic units of nucleic acids made from pentose, the 5-carbon sugar. And on the 5-carbon sugar, you have a nitrogenous base and a phosphate group. That gives you the structure of a nucleotide. Here we are looking at what we refer to as a dinucleotide. So the first nucleotide is having nicotinamide as the nitrogenous base, which is bonded on the first carbon of ribose. Now, remember also that when we were looking at nucleotides, we saw two forms of the sugars. We had the deoxypentose, and we also saw the oxypentose. And this is the oxypentose, which you find in ribose. If this hydroxyl group was replaced with just hydrogen, that will become the deoxypentose, which you find in DNA. In this case, of course, you don't find the nicotinamide nucleotide in nucleic acids. You don't find that. Here we have this special nucleotide, which is having the nitrogenous base being nicotinamide on the pentose sugar ribose, and there is a phosphate group also on the carbon number 5. Then we have a second nucleotide, the nitrogenous base being adenine. This you have seen before. So you have adenine and you have the sugar and then a phosphate group also on the carbon number 5. And here we see that there is that phosphodiester bond that is formed between these two phosphate groups. Now you remember also that when we were studying the structure of nucleic acids, we talk about the bonds which exist between the nucleotides in a nucleic acid chain being phosphodiester bonds. But there is some difference between the phosphodiester bonds which you find here and those that you find in the nucleic acids. And that is, in the nucleic acids, you find that the bond, that phosphodiester bond which you find is actually between carbon number 5 and carbon number 3. If you remember that. And actually, it was only with one phosphate group, just one phosphate group from carbon number 5, forming such a phosphodiester bond between carbon number 5 and then carbon number 3. And that's how come you in nucleic acids you have a 3' end and you have a 5' end. But here you see that the phosphodiester bond or phosphoanhydride bond, which is being formed here, is actually between carbon number 5 and carbon number 5. So this is not the same as the phosphodiester bond, which you find in nucleic acids. So, this is being referred to specifically as a phosphoanhydride bond. Right, so you find two nucleotides which are bonded together. And so this is a dinucleotide, and that is why you see the name being nicotinamide adenine dinucleotide. Nicotinamide coming from this base, and adenine, the second base, and a dinucleotide. Once you see dinucleotide, of course, you know that involves sugar and phosphate group. And so it tells you that there are two nucleotides that are bonded together, and the bases of those two nucleotides are nicotinamide and adenine, respectively. Now, you have seen in many chemical reactions the nicotinamide adenine dinucleotide being represented as NAD, or sometimes you see NADP. Sometimes you see NADH, sometimes you see NADPH. We will go into all of that for you to have a better understanding of the differences. But you see on this carbon number 2, in place of this hydroxyl group, if you put a phosphate group here, if a phosphate group binds on carbon number 2, replacing this hydroxyl group, that will give you what we refer to as the nicotinamide adenine dinucleotide phosphate. Right. So we'll go ahead and we see the differences between NAD+, which is the oxidized form of the nicotinamide nucleotide, and NADH, which is the reduced form. We'll also see the difference between NADP+, which is the oxidized form of that phosphorylated analog. If you have a phosphate group on carbon number 2, replacing this hydroxyl group, that phosphorylated group makes it NADP. If it's NADP+, that is the oxidized form of the coenzyme. NADPH becomes the reduced form of that coenzyme. Right. Now, what is the function of these coenzyme, the nicotinamide nucleotides? Now, the nicotinamide nucleotides are electron carriers. So you find them in oxidation reduction reactions. So for instance, like reactions in which you have transfer of electrons and together with transfer of protons, you find these nicotinamide nucleotides as the carriers of these electrons and the protons from one molecule to the other. So you see this coenzyme, again in an oxidized form, carrying an electron. So if it carries an electron and then some proton, what happens is you now have, so the NADP+, you see you have a plus on this nitrogen. We'll come to what actually this plus also stands for. And the reduced form is now carrying lone pair of electrons. So we have that carrying that electron, and you have one of these hydrogen, these protons is attached on this carbon number 4. And you see, we'll also come to what this means. You see one of this hydrogen is pointing towards you. Remember your stereo chemistry. When you see this bond, it means that the, the atom is pointing towards you. And when you see these dashed lines, it means that hydrogen is pointing away from you. And you see this being referred to as A side, which is the side which is towards you, and the B side, which is the side which is pointing away from you. We'll come to what this means, what the A side, the B side means as far as stereospecificity is concerned. And you see that the NADH, when you have the hydrogen atom, the proton being accepted on the nicotinamide base that is in that dinucleotide, you refer to that structure as the reduced form, NADH, the reduced form of the coenzyme, the nicotinamide adenine dinucleotide. And when you have NAD+, you refer to that as the oxidized form of that coenzyme. Right. Now, the function of nicotinamide nucleotides is such that it is, as we said, carrier of electrons or protons from one molecule, carriers of electrons and protons from one molecule to the other. But the difference between NAD and NADP in this function is that when you have in an oxidation reaction, you find that mostly NAD+, the oxidized form of NAD, acts in that reaction, in reactions of oxidation. And that is because the NAD+ accepts hydride ions. And as it accepts hydride ions from substrates, the substrate then gets oxidized whilst the coenzyme gets reduced. So this is a coenzyme. It's in its oxidized form. When it accepts the hydride ion, it gets reduced. And once it is a coenzyme that is getting reduced, then definitely the substrate gets oxidized. Now, similarly, for reduction reactions of reduction, you find that it is the phosphorylated form, the reduced form of the phosphorylated form, which acts generally in those reactions. So in the process of reduction, you find NADPH being a donor of hydride ions, which are accepted by a substrate. And in that process, the NADPH now gets oxidized to NADP+. And as it gets oxidized to NADP+, the substrate is then reduced. What it means is the substrate rather accepts the hydride ions from NADP to get reduced. So functionally, these are the differences between the nicotinamide adenine dinucleotide, which usually you refer to as NAD or NAD, and the phosphorylated form, which is NADP. So you see the functional difference between these two. The NADP+, which is generally responsible for oxidation reactions, and NADPH, generally responsible for reduction reactions. All right, of course, these reactions generally occur in the powerhouse of the cell, which is the mitochondria. Either it is in the mitochondrial matrix, or is occurring maybe in the electron transport chain in the mitochondrial membrane, where you have the production of cell energy in the form of ATP. Now, remember, though, there's a small note here that, the plus sign, which you see in these abbreviations like NAD plus or NADP plus, really, it's not indicating a net charge. That's not what it is. It is only indicating that the molecule is in its oxidized form, but not a charge. And when you see NADH or NADPH, it is denoting that that coenzyme is in its reduced form. So that plus sign you see is indicating the form, the oxidative form in which the molecule is, but not a net charge. All right. Now, we see some specific example of the reaction in which, reactions in which the nicotinamide nucleotides are involved in. And in this case, we are looking at alcohol metabolism, where you have, this is ethanol, and ethanol is being oxidized, first of all, to acetaldehyde. And you see the coenzyme NAD+, getting reduced to NADH in that process. That is an oxidation reaction. You have ethanol that is being oxidized to acetaldehyde, and the enzyme that is acting in that process is alcohol dehydrogenase. And the coenzyme to alcohol dehydrogenase is NAD+, which gets reduced to NADH. And the reaction goes further. From acetaldehyde, you have further oxidation to acetic acid. Again, in that process, you have the enzyme alcohol dehydrogenase. In some biochemistry books, this is distinguished. So you have the first, which is, because the substrate is alcohol, the enzyme there is being referred to as alcohol dehydrogenase. And in the second reaction, because the substrate is acetaldehyde, you find the enzyme here being referred to as acetaldehyde dehydrogenase, because that's the substrate that's getting oxidized to acetic acid. And in both cases, the coenzyme is NAD+, which is getting reduced to NADH. So you see that specific example in which you find this nicotinamide nucleotide acting as a coenzyme to the enzyme alcohol dehydrogenase. Of course, you may have seen a lot of reactions, a lot of biochemical reactions with NADH, NAD, maybe you didn't pay attention to that aspect of the reaction. You may just be looking at the substrate and then, or the reactants and then the products, but not necessarily at the enzymes and then the coenzymes which are acting. But this time we are focusing on that, on the coenzymes and the enzymes that are acting in those biochemical reactions, so that you can have a better understanding when you read next time these biochemical reactions and you see these enzymes and coenzymes acting, you will know specifically what their functions are in those reactions. Now, we talk about the stereospecificity of the enzymes that have these nicotinamide nucleotides as their coenzymes. I mentioned that we'll come to what it means for the hydride ion to be bonded on the A side, which is the side of the nicotinamide ring, which is pointing towards you, and what it means if the hydride ion points away from you, which is referred to as the B side. Now, you remember we talked about different forms of specificity of enzymes, and we refer to some absolute specificity, root specificity, bond specificity, and all of that. Now you are looking at a different form of specificity of enzymes being referred to as stereospecificity of the enzyme, and particularly based on NAD, nicotinamide adenine dinucleotide spatial orientation, such that you have enzymes that are going to be active with the coenzyme nicotinamide adenine dinucleotide that have the orientation of the nicotinamide base that is having its A side pointing, too close to the enzyme for that, transfer of electrons and the hydride ions being bonded onto that carbon number 4. So you find that here you see an enzyme, isocitrate dehydrogenase. This is an enzyme which you find in the citric acid cycle, what you refer to as the Krebs cycle. And of course, as the name suggests, isocitrate. Isocitrate is one of those intermediates you find in the TCA cycle. Dehydrogenase is an oxidoreductase. And in that process of oxidation of isocitrate, you have the coenzyme, which is acting being NAD+. And that NAD+ has stereospecificity to the nicotinamide ring, which is accepting the hydride ion on the A side. So which means if NAD+ was present, but it was the B side, which was oriented towards the electron transfer or the hydride ion from the substrate, that NAD+ will not activate the enzyme isocitrate dehydrogenase. It will not. If the B side was what was, if the orientation is that it was the B side which was close to the enzyme to receive hydrogen ion, it will not activate the enzyme. That reaction will not, proceed. It is only when you have the A side, present that you have. So it's like a switch, on and off, on, off. If the A side is pointing towards, that orientation is there to accept the hydride ion from substrate, then reaction goes on. If it is the B side, reaction does not go on. That is stereospecificity. And you see that for other enzymes. Alpha ketoglutarate dehydrogenase is also an enzyme that we looked at earlier. It's also found in the TCA cycle. And the coenzyme NAD+, again, this time around, stereospecificity to the B side. Then you have glucose 6-phosphate dehydrogenase, what you commonly refer to as G6PD, and that has the coenzyme NADP+. It's also an oxidative reaction. But this one, uses the phosphorylated NAD as the coenzyme, and it has stereospecificity to the B side of the nicotinamide ring. Malate dehydrogenase, you also find in the carbohydrate metabolism, stereospecificity to the A side. You have glutamate dehydrogenase that will be found in amino acid or protein metabolism, either NAD+ or NADP+, stereospecificity to the B side. Glyceraldehyde 3-phosphate dehydrogenase, you find also in carbohydrate metabolism, has the stereospecificity to the B side. Lactate dehydrogenase. And the last, which we just saw an example of, the alcohol dehydrogenase. You see both of these enzymes have stereospecificity to the A side of the nicotinamide ring on the coenzyme NAD. So you see that stereospecificity as, a separate kind of specificity, which you can add to what we have learned earlier about enzyme specificity. So, enzymes that, will catalyze reactions based on this specificity might be grouped as, for instance, A-type, an A-type enzyme. If you have the nicotinamide nucleotide being a coenzyme to it, the enzyme may be classified as an A-type if the stereospecificity is to the A side of the coenzyme, and it will be classified as a B-type if you have that hydride ion transfer or proton transfer onto the B side of the nicotinamide ring. Right. Now we look at, so here, the nicotinamide, this, you realize we said we were getting it from vitamin B3. We talk about thiamine coming from vitamin B1. Now we see this is another form of nucleotide, which is referred to as flavin nucleotides. These two you would have seen in reactions, FAD. Sometimes you see FADH, for instance. And this is coming from vitamin B2. So we jump to B3, then now we have come to B2, vitamin B2. And this is riboflavin. Vitamin B2 is called riboflavin. And we have the dinucleotide having this structure where, again, you have adenine on the 5-carbon sugar with the phosphate group on the carbon number 5. So again, you have adenosine. So that adenosine is bonded onto a flavin mononucleotide. And the flavin mononucleotide, again, so actually, this is the linear structure. Remember, first year biochemistry, we looked at the linear structure of monosaccharides. So if you look at these carbons, 1, 2, 3, 4, 5, these are 5 carbons. So this is also a pentose. You have a 5, carbon sugar with a phosphate group on carbon number 5. And then you have this, flavin structure, which is what we refer to as isoalloxacin ring. These are some 3 6-sided rings fused together. If you were having just one phosphate group on this, of course, that will be a flavin mononucleotide, which is abbreviated FMN. Now you have these two nucleotides together. So that's a dinucleotide. You have a flavin adenine dinucleotide. Flavin, so flavin being that nitrogenous base. And then you have adenine being also a nitrogenous base. And these two structures are nucleotides, and both of them are bonded together by a phosphoanhydride bond. So again, the phosphate group on carbon number 5, here too, phosphate group on carbon number 5, and there is that, phosphoanhydride bond formed between these two phosphate groups to give you the full structure being a flavin adenine dinucleotide. Now you see that flavin adenine dinucleotide here also, can have some acceptance of hydride ion, or call it a proton, and some electrons. And as that happens, if you have the flavin, this flavin nucleotide accepting one hydride ion, what you have is that you have what we refer to as semiquinone, and you see it being represented as FADH. That is only one H. With some dot like a radical, or if it is flavin mononucleotide that is accepting, that hydride ion also, So you have accepting a hydride ion and, one electron, for instance, and that gives you a semiquinone. If it is accepting 2, so that is if this first semiquinone accept more, then you now have two hydrogens being accepted, two hydride ions being accepted, and you now have FADH2. That is fully reduced flavin nucleotide. So you see the difference between NAD and FAD is that FAD can take up to 2 hydride ions, can accept more, but NAD can accept only 1. Now here, what happens is that you have, so you can clearly see that both of the NAD and then FAD are involved in oxidative reduction reactions, accepting hydride ions and electron ions. With NAD, we mentioned that they are responsible for the transport of these electrons from one molecule to the other. So it can be, 2 electrons in the form of, 1 or 2 hydrogen atoms, to form that reduced substrate. Now, you see that in the case of the flavin nucleotides, these flavin nucleotides are tightly bound to the protein. You see the protein, which is the enzyme, that you have these organic compound being bounded to as a prosthetic part to that protein. That protein is then termed a flavoprotein. It's termed a flavoprotein because you have these flavin nucleotides that are bound to them to make them what we saw as what? A hollow enzyme. You have a coenzyme bonded onto an enzyme. You have a hollow enzyme. That hollow enzyme is being referred to as a flavoprotein. In this case, you have the flavin nucleotides bonded tightly to this. So the flavin nucleotides do not move from one molecule to the other, transferring these electrons. Rather, they are embedded. They are embedded with the enzymes so that they have that acceptance of the hydride ions, electrons, onto them. They transfer them not by the movement of themselves. They can be transferred not by the movement of themselves. I mean, the coenzymes themselves, no, because they are tightly bound to the proteins that they are bonded to form a hollow enzyme. Now you see that, some of these flavoproteins will have some additional, call it this time around a cofactor. So they can be bound to some inorganic metallic ions like molybdenum or iron. So even though they are acting as coenzymes, they may also require some cofactors so that you have that full structure, the full structure of the enzyme becoming active. You find enzymes such as acyl-CoA dehydrogenase, which you find in lipid metabolism, having flavin nucleotide as their coenzyme. Others, such as dihydrolipoldehydrogenase, is also found in lipid metabolism. Succinate dehydrogenase in carbohydrate metabolism. Glycerol 3-phosphate dehydrogenase, you find also in lipid metabolism. Thioredoxin reductase, I think you find this in protein metabolism. You find NADH dehydrogenase, that is, in the oxidation of this molecule, NADH, which is a coenzyme on its own, forming a complex. You find flavin mononucleotide acting as a coenzyme. And you find glycolate oxidase also in carbohydrate metabolism, making use of flavin nucleotides as coenzymes. Now, another interesting part of flavoproteins is that they can be acting as what is referred to as cryptochromes. And These are responsible for what we refer to as the circadian rhythm in mammals. In other words, what we call biological clock. So even in plants, you find this. So for instance, in the morning at sunrise, you see plants opening up, their leaves opening up or their flowers opening up at sunrise. And at sunset, you see the leaves of the plant closing, the flowers closing. That is what is referred to as circadian rhythm. So for mammals, for instance, when it's sunrise, when it's the atmosphere gets clear, the mammal should be awake, should wake up from bed, from sleep, because now there is enough light that could enter into the eyes, and that would trigger various hormones, and then you have that activity coming into the body of the mammal. When it is late in the night, somewhere around 10 o'clock there, but when it gets dark, when not enough light is entering to the eyes, the mammal should fall asleep. That is a circadian rhythm. Now in plants, these cryptochromes actually will have this mediation of the effect of visible light, specifically blue light, on plant development. Either the plants should close up, open up, when photosynthesis should take place at its full scale, when photosynthesis reduces, sometime late in the night, maybe photosynthesis and respiration becomes equal. All of that is being regulated by these flavoproteins, being referred to as cryptochromes, because they are receptors of light. You find these flavoproteins also acting as photolysis. So we saw, one of the classes of enzymes being lysis. So, and those are responsible for what? Breakage of bonds, in compounds. And you find that in prokaryotes and eukaryotes as well. And these photolysis will act based on absorbed light. So they get energy from absorbed light, and especially acting in the reparation of DNA. So DNA that has some defects, chemical defects in DNA molecules, can be repaired by this process where flavoproteins act as photolysis, removing the damaged parts of the DNA and then actually incorporating back new nucleotides for the reparation of these damaged DNA molecules. Now, the next coenzyme is coenzyme A, which you have seen in many chemical reactions. Here also we see that the structure of the coenzyme is made up of adenosine, where you find that this is a phosphorylated adenosine. Again, here you see adenine as the nitrogenous base on that ribose sugar. But on this ribose sugar, you have a phosphate group on carbon number 3. And so we can say that is a 3' phosphoadenosine. Then you have on this carbon number 5, again, you have 2 phosphate group. That's a diphosphate. And then we see there is a structure which is shaded in magenta, referred to as pantothenic acid. Now, pantothenic acid is vitamin B5. Vitamin B5. So again, we see another vitamin B before we have the last end of the structure being beta-mercaptoethylamine. So that tells you that is some form of an amino acid, some residue of course, not any of these amino acids which the 20 amino acids which we find in proteins. This is a different kind of an amino acid which you don't find in proteins. In fact, you may find beta mercaptoethylamine only after you have breakdown of coenzyme A, because this is the full structure in which you find that part of the coenzyme. And here, the reactive part of this coenzyme is the sulfur hydride, group, which is referred to as the thiol group. Again, you see the term thiol, the sulfur hydride group is what is referred to as the thiol group. That is the reactive part of the coenzyme. So this whole structure, the reactive part is this terminal end, sulfur hydride end. And you can say this looks more like one of maybe a residue from one of those the coenzymes, having sulfur in them. Maybe cysteine, for instance, maybe cysteine, yes, maybe cysteine. So you have SH at the end, which is the reactive part. So that if this whole structure, this whole, apart from the sulfur end here, is represented by S, OA, an acetyl group attached, replacing that hydrogen, will now give you the structure of acetyl-CoA, which you have also seen. It's like the acetyl group carries the coenzyme A in chemical reactions. So you see why you can't be writing this structure every time. This is so, it's complex, big, and so you see that a lot of time you see this being written as CoA. If you add an acetyl group to it, you have acetyl-CoA. And it's simpler to write it this way because you have, some phosphoadenosine diphosphate bonded onto pantothenic acid, which I said is vitamin B5, before you have some beta mercaptoethylamine. So that's about the structure of coenzyme A. And you find coenzyme A in many chemical reactions. If you go into the TCA cycle, the first reaction you see there, you see coenzyme A, you see acetyl-CoA actually, having some, reaction with oxaloacetate, giving you isocitrate. Right from the beginning, you will see coenzyme A there. If you go into protein metabolism, lipid metabolism, all these metabolic reactions, even in nucleic acid metabolism, you find coenzyme A acting in those reactions. Now, we see also biotin. And biotin is also another, vitamin B, which is vitamin B7. Vitamin B7. And we see that the reaction in which biotin is involved is that of carboxylation. This time around, not decarboxylation, but rather carboxylation. So we see the carboxyl group in red, which is from the bicarbonate, is transferred onto this substrate pyruvate to now have oxaloacetate. So you have this carboxyl group now attached, in place of this CH3, you now have CH2, one hydrogen is gone, and you have the carboxyl group, conjugated on that pyruvate residue to now have oxaloacetate. And you see this sort of reaction in carbohydrate metabolism. And the enzyme which is involved in that reaction is pyruvate carboxylase, the coenzyme being biotin. Of course, with some expense of energy, ATP is being expended. So some phosphorylation occurs. So the phosphorylation of the enzyme pyruvate carboxylase as a form of activation. And so you have, ADP left and the coenzyme being biotin in that reaction. And you see the structure of biotin, two 5-sided rings that are fused together. You have some sulfur, and then you have some C double bond O groups and some functional groups present here. And you have some carboxylic acid group attached, a valerate, which is attached on that two 5-sided ring fused rings together. Now, an interesting thing that you should pay attention to is the fact that biotin deficiency can be caused by the consumption of raw eggs. So raw egg whites have the protein avidin, and this avidin can bind very tightly to biotin, preventing its absorption in the digestive system. So if you have vitamin B7, which is biotin, its absorption being prevented in the digestive system to go into the circulatory system, to go to all the tissues, of course, that's going to result into deficiency of these vitamin B7. And of course, it means deficiency of these coenzyme. And that, of course, can result into malfunction of the enzymes that you have, this vitamin acting as a coenzyme too. Now, we see the mechanism of action of biotin. So if this is, biotin carrier protein will be that enzyme, it will be an enzyme that you have the biotin as a coenzyme being bonded onto. And you find that you have, so the reaction which we saw earlier, the bicarbonate. So we saw here, this is a bicarbonate from which you have the carboxyl group, which is transferred onto the pyruvate, residue to give you oxaloacetate. How will that occur? So in the first case, so this is the biotin molecule on the enzyme, which is being referred to as the biotin carrier protein. And you have the enzyme which is acting in the reaction being what? Biotin carboxylase, which is responsible for the carboxylation of biotin. So the carboxyl group is carried on biotin, which is the coenzyme. First of all, there is an expense of energy there. And then in the second step, you now have acetyl-CoA with trans-carboxylase being the enzyme acting in that reaction. And this carboxyl group is now transferred onto acetyl-CoA to give you malonyl-CoA. So it's a similar thing which occurs with the transfer of the carboxyl group onto the pyruvate residue to give you oxaloacetate. This time around, it is onto acetyl-CoA to give you malonyl-CoA, which you find in lipid metabolism. So when we come to lipid metabolism, you will see malonyl-CoA acting in that reaction. So this is an example of the mechanism of action of biotin carrying carboxyl group from one molecule, the bicarbonate, onto a substrate, acetyl-CoA, to give you malonyl-CoA. So generally, you see biotin taking part or participating in reactions of carboxylation, transfer of carboxyl groups. C double bond O, CO, sometimes being written as CO2. Not necessarily, this does not mean carbon dioxide. It means the carboxyl group being transferred from bicarbonate onto a substrate. Right. Now we move to what we generally refer to as folic acid. Here we call it simply folate. And that is coming from, this is also one of those vitamin Bs, which we refer to as vitamin B9, vitamin B9 as folate or folic acid. And we see this is also coming from 3 different molecules. And you see that as glutamate. So glutamate, that is glutamic acid, one of those amino acids that you saw with an extra carboxylic acid group in the side chain. So you see that glutamate or glutamic acid. Then we see para-amino benzoate residue. I mean, that is also, referred to as para-amino benzoic acid. That itself, para-amino benzoic acid or para-amino benzoate, is also referred to as vitamin B10. So vitamin B10. Then we have the 6-methylpterine. Now, 6-methylpterine also is coming from what is being referred to as vitamin B11, which is, usually, so like this whole structure, sometimes you see vitamin B11 being referred to as teryl heptaglutamate. So you see that there is that connection between vitamin B10, that is the para-amino benzoate, and then you have that vitamin B11 factor, together with this amino acid, glutamic acid, being. But this whole folate, the folic acid is referred to as vitamin B9. Of course, some of these vitamins, it was later, some of them were later discovered not to be vitamins per se. So they have whole history. I can't remember whole history of all of these vitamin Bs you can have, from vitamin B1 up to about vitamin B20, and later some of them were seen to be really not vitamins, like the nitrogenous bases like adenine, some structure like choline, which we saw in the structure of acetylcholine, carnitine, which you find in the metabolism of, I think, fatty acids, being referred to as vitamin B4, for example, and later were discovered as not being vitamins, but different compounds on themselves. Vitamin B13, for instance, was orotic acid, which was, found to be a byproduct of metabolism of nucleic acids. We'll come to see that as we go on. Even cobalamin, for instance, which was also considered to be vitamin B12, or I think still they refer to it as vitamin B12, cobalamin. Right. So the whole lot. If you're interested, you can go read on vitamin Bs, and you see a lot of these vitamin B. Some of them are no longer being referred to as vitamins. Some of them are still being referred to as, and then many of these vitamins with different functions. Some of them, the functions not well understood, and all of that. So let's go on. We see this folic acid, vitamin B9, and in the structure we have seen these various components: the glutamic acid, the para-amino benzoic acid, the 6-methylpterine ring, and you see here too we have the nitrogen 5, N5 and N10 in blue, and that is because those are the reactive portions of the whole structure of folates. Now, the functions of folic acid. Generally, folic acid is, they are involved in what's referred to as the transfer of single carbon groups. So single carbon groups like methyl group, like CH3, or methylene group, like CH2, or, the formyl group, that is the aldehyde group, like C double bond OH. The transfer of these groups from one molecule to the other, which actually results into modification of these groups. And usually that is in biosynthesis, anabolic metabolism, the build-up of bigger molecules from smaller molecules. And these reactions are usually being referred to as folate-mediated one-carbon metabolic reactions. And you also, know that folic acid is generally, prescribed for pregnant women. And that is because folic acid is very much important in reactions that are involved in cell proliferation. So the growth of cells and the specialization of cells. So at that age of the fetus, where you have the fetus developing in different stages, where that fetus develops into a full human being, there is the need for adequate amount of folic acids or folates present, so that, especially for the brain, that development of the brain. So the folic acid being prescribed for pregnant women actually is to reduce the risk of what is referred to as neural tube defects. That is brain damage. That is, defects in brain formation of offsprings. That is why you have this folic acid being prescribed for these, pregnant women. All right, so you can see that generally we have been talking about vitamin Bs, different vitamin Bs acting as coenzymes, and that actually explains, why a lot of times when you have patients, going to a hospital, for one ailment or the other, and you have prescriptions being given to them, you find that to specific prescriptions, you have vitamin B complex being also added to these prescriptions. And that is because the vitamin Bs are needed as coenzymes to the enzymes that are needed for the metabolism of these medical or medications that are being given to the patients. No, drug metabolism in the liver requires, the action of various enzymes. These substances which are being taken in, these drug substances, are xenobiotics, substances from the external environment taken in, and they require, a lot of, enzymatic action for their metabolism. And if, a system that is already ailing may not be having, enough capacity to, undergo the metabolism of these drugs, you need to have these coenzymes in place, as in the form of the vitamin Bs, to actually boost the drug metabolic system, or the process of biotransformation of these drugs, to actually have the best outcome from these drugs. So that is why you see that a lot of these vitamin Bs are, the vitamin B complex is being given in addition to many of these medications. Sometimes, you are told it helps with the immune system and all that. It's all about biochemical reactions that are occurring in the body, and it's to do with enzymatic reactions and how these vitamin Bs act as coenzymes to the enzymes that are involved in those enzymatic reactions. So generally, we have looked at thiamine pyrophosphate, nicotinamide nucleotides, flavin nucleotides, coenzyme A, and then we look at biotin, and we also look at folate. All of these being some form of vitamin Bs. Of course, coenzyme A containing pantothenic acid, which is vitamin B5. So that brings us to the end of this lecture on coenzymes. The next time we will meet, we'll start to look at metabolism of biomolecules like carbohydrates, move on to proteins, we move on to lipids, move on to nucleic acids, on and on and on, one after the other. So now, anyone with any question, any contribution, anything you want to say, I mean, you can raise your hand, I'll call your name, and you unmute yourself, and then you can speak to our hearing. Yeah, so if you have any question and you have anything to say, the floor is open. You can speak. So this lecture is being recorded. So when we finish, I will send the recording to Wisdom, so Wisdom can share it with you. I'll also share the slide with him, so he will share it with you in your book. Yeah, so anything, the line is open now, so you can talk. Well, okay. Yes, Ibrahim, you can unmute yourself and speak.
Sir, please, is there, like, when we take the human body like this, do we have like to say enzymes that are natural? They are already natural made in the body, or you have to take proteins before they can be synthesized, since proteins are enzymes are made from proteins?
Okay, that's a very interesting question. Now, first of all, enzymes are endogenous compounds. So what I mean by endogenous compound is that these are compounds that are existing in us. Okay, the enzymes are there. It's not that it is these enzymes are being taken from, say, food, external food into us. That is one part. Okay, but now proteins that we find in us, proteins which some of them become the enzymes, are synthesized. We synthesize proteins. I mean, the proteins which we refer to as non-essential proteins, right? Non-essential proteins, meaning that we are able to synthesize them. And we have essential proteins. Actually, we refer to essential amino acids and then non-essential amino acids, so that we are able to synthesize some of the amino acids and we are not able to synthesize some of the amino acids. Now, the amino acids that we are able to synthesize, even in the body, if we have some proteins being taken in, right? We have proteins taken in from food. That food will have to be broken down into amino acids. And it is the amino acids which we get from the food that we take in, which is used in the synthesis of proteins in us. Now, here is the thing. You see, we talk about metabolism as broken down into two branches, catabolism and anabolism. Now, catabolism is the breakdown of the food that you take in, a breakdown of those bigger molecules. So in this case, the breakdown of the proteins that you take in into amino acids is a process of catabolism. Now you have amino acids. And in that case, you have some amino acid pool. And from that amino acid pool, you now can synthesize proteins from these amino acids. So yes, in some breath, you need amino acids from the food that you take in order to synthesize the proteins that you need. Not all of the amino acids are taken from food. Some of them can be synthesized in the body. So, I mean, your question is actually a question of metabolism of proteins, where you take in some proteins and it's broken down into amino acids. And those amino acids are then used to synthesize the proteins which you need. And some of that protein becomes enzymes in the body. I hope that makes it clear to you.
Yes, sir.
All right.
Thank you.
Okay. So Sylvia is saying, can we get previous one, which is what? Inhibitors. Previous what? I think with the inhibitors, I thought I shared the recording with unless of course it was one of those lectures we had which we couldn't record. But any of the lectures which we were able to record, I've shared them. And I have shared the presentations too. All right, any other? So I think, Ibrahim, when we come to protein metabolism, for instance, you have a better understanding of what I just said, because I think they will be looking at, amino acid pool, the sort of, the amino acids that we need for synthesis of proteins, and things like that. You'll see that in protein metabolism. So it's interesting. You take protein from food. So for instance, if you take protein from some meat or from milk or from even some plant food, remember those proteins that you have taken from those foods are not the same proteins which you need. And so they have to first of all be broken down into amino acids. And even in that case, those proteins which you have taken in, they are non-functional because you have cooked them, for instance. So they are not in their functional state. They are denatured. And so it is when you take them in, they break down, and then you can get amino acids from them. And then you have that process of protein synthesis. If you remember, protein synthesis from the process of translation. So going back to your molecular biology, maybe from HND, where you did the process of replication, and then you process of transcription, and then you look at process of translation, which is a synthesis of proteins from messenger RNA. And those proteins, some of them become enzymes, some become hormones, some become some neurotransmitters, and different other things in the body. Yes, any other question? We have 10 minutes to 7. If there is no question, okay, so Sylvia is saying recording. So I don't know if we recorded the lecture on inhibitors, but if we did record, then I have shared them. But if we didn't, then it means it's not available. But the slides are there; they are available. I've shared them. All right. So I've seen some people are already leaving. So if maybe their data is finished or anything, or maybe they themselves they are just tired. So if there is nothing else anybody wants to say, then so we meet. I hope that the next time, by the time our next lecture, I don't know when the exam is ending, so that maybe in our next time meeting it will be face to face, for instance. I don't know exactly. Maybe by the time the week ends, that will be clear. All right. So all the best. Have a good evening till we meet the next time. Bye for now.
Bye.